Welcome back to our speaker number 3 for our session today of the 2021 DOE Summit. We have with us Stephen Mullen. He was born and raised in the suburbs of Minneapolis, Minnesota. He attended the University of Minnesota and completed a bachelor's in biology in 1995. He went on to study cryobiology and reproductive physiology and finished his PhD in the College of Veterinary Medicine at the University of Missouri-Columbia in 2007. He has been employed in industry since 2008 and continues to work in the field of cryobiology and assisted reproduction. He's presently the team lead and senior scientist at Cook Medical in Bloomington, Indiana. So welcome, Stephen.
Well, thank you, Sherry, and thanks so much for the invitation to come and talk today at this summit. I attended last year's, and it's something I quite enjoy. And as you and I have talked about, statistics is kind of an interesting topic for me, something sort of a side topic I really enjoy. So I've done my best, and this is my first adventure into actually a response surface methodology project. So I've learned a lot over the past 2 years, and would say definitely that Stat-Ease has helped with that process. So a disclosure statement here, I'm employed by Cook Medical, and we do manufacture and distribute services and products for the IVF industry. And so what I'd like to do today is just discuss a bit about the project, and I am going to be going over a bit more background in the field of assisted reproduction, because I know that the audience here is not the typical audience that I would talk to, which would include people who are familiar with the field of ART. And so I will be talking about the background and an introduction to the problem. And as soon as I can get my things to work here, and then give a brief overview of the preliminary experiments I conducted to identify some key variables associated with the problem we're going to talk about. And then the approach to the multi-factor design that I designed with the help of Stat-Ease, or design expert, excuse me. And then the model that came out of that project and the analysis of that model, the accuracy of the predictions and interpretations of this model, and finally summarize and open it up for questions. And so to start with, I'd like to just briefly talk about infertility. And this is a topic that most people are not very familiar with unless it directly affects you. And it, in fact, indirectly affects quite a lot of people. It's estimated that about 15% of couples face infertility of some level. And there's some facts on the right here that I took off of the reproductivefacts.org homepage. And some of the things to talk about is it affects about 6.7 million women in the United States annually. That's about 11% of the reproductive age population. And it doesn't only affect women, which is something, it's probably a bias that many of us at least in society, have had over the years. It affects men equally as it does women. And about 40% of infertile couples, the male partner is either the sole cause or contributing cause of infertility. And so it's really a problem for both genders. And IVF has really come along quite strongly in the past couple of decades, whereas in 2019, about 77,000 babies were born in the US from ART. And in vitro fertilization and embryo culture are key components of these processes. And finally, for those who suffer from infertility, it's very emotionally distressing. And many people suffer depression when they're diagnosed with infertility, and it's really a big problem for people. And so I hope all of us would take this into consideration. So these are some data that I took off of the Society for Assisted Reproductive Technology website, showing how the number of IVF cycles has increased quite dramatically over the past couple of decades, as has the number of live births resulting from these treatments. And the processes and the technologies involved have been evolving over time. But the one thing to point out is that in vitro fertilization and consequent embryo culture has been part of this process since the beginning and continues to be a key component within IVF. And so a question I'd like to pose and then answer for you is, how do we actually culture embryos in vitro before they're transferred back to a recipient? And historically, starting back in the early 1960s, it was determined that culture media could be formulated and made into microdrops like we see in this top left picture. And then one of the benefits of that is you can find the embryos quickly under a microscope and observe them as they develop. Now, because they're in this microdrop format, we have to worry about evaporation of the media, which can happen quite quickly in these small volumes. And so these drops are covered with a mineral oil, as is shown in this dish on the right. And then those dishes with the mineral oil covered embryo drops are cultured in a traditional cell culture incubator. And these pictures down here show what the embryos look like as they develop. Initially, we start with an unfertilized human egg, or oocyte, which is shown here on the left. And they're pretty big cells as far as cells are concerned. They're about 125 micrometers in diameter. Now, if the egg gets successfully fertilized by a sperm, either through traditional in vitro fertilization, which would occur in a microdrop like this, or through a process of directly injecting a sperm into the egg, which is shown by this picture here. And just to give you a sense of the scale difference between these cells, this little speck right here inside this pipette is the sperm cell. And that cell in this process would be directly injected into the egg using this glass micropipette. And if everything goes according to at least the desires, you would have normal fertilization and then the progression through preimplantation development, as can be seen in these pictures here. And this process would take place over about a 5-day period in the human. Whereas if things go well, you would end up with an expanded blastocyst, which we see in this final picture here, which is an embryo that has a large number of epithelial-like cells surrounding this hollow structure. Those cells would eventually become the majority of the placenta. And if you can see here in the center of this, there's a mass of cells, which is generally referred to as the inner cell mass. And the cells within this structure become the embryo proper. And just parenthetically, this structure here, this inner cell mass, is the source of the embryonic stem cells that have been in the news so much in the past couple of years. And so you can imagine how important it is for this process to occur normally for couples who are undergoing infertility treatment. And as such, it is really incumbent upon those of us in the field to do this as best we can. So I'm going to start to define the problem that caused this project to be initiated. And for a little bit more background, you probably can understand how these processes would have evolved as our knowledge has evolved over the past 30 years. And methods to culture embryos have changed to some degree over the past few years. And some of these changes include the use of non-humidified incubators. And the use of non-humidified incubators has come along in sort of concurrently with the use of time-lapse imaging during the incubation process, which allows the embryologist to monitor the progress of the embryo development and see and observe and sort of time out some of these key time points within this 5-day period. Now, because we were using this oil layer over the media drops, it was assumed that if we cultured these cells in a non-humidified incubator, that oil layer would protect the media from evaporation. And of course it does to a significant degree, but what we found out over the past couple of years is it does not completely eliminate water loss from these drops, which is a bit surprising. And when water gets lost from the drops, what happens is the total concentration of solutes in this media changes and it increases. That increases the osmolality of the extracellular medium, and that results in water loss from the cell to compensate and try to reach an equilibrium state between the cell and the extracellular medium. And what that can do is not only changes the chemical concentration of the media itself as well as the pH, but it also will change the intracellular components of the embryo also. Now, there are some means by which embryos can compensate for these changes in osmolality, but what has been shown by different investigators is that despite the ability of the embryos to compensate, there does seem to be an effect, a detrimental effect on culturing embryos in these non-humidified conditions. And I'm just going to focus on one of these studies here and one image from this particular study. And I think this is the earliest study that I found that really highlighted this problem. And what they showed was when they cultured in humidified or non-humidified conditions, there was a decrease in the non-humidified condition of the quality of the embryos at both day 3 and day 5, and also the pregnancy rates that associated from embryo transfer. And this sort of set off a lot of alarm bells within the community, and people began to ask the question of whether these changes in these culture systems really are sort of neutral, if you will, or if we really are causing a problem we didn't think about. And so a lot of other people began to think about this and began to investigate different factors within an embryo culture system that might contribute to this change in osmolality that is seen when embryos and culture media are cultured in a non-humidified environment. And perhaps not surprisingly, people would perform the so-called one factor at a time approach. And there were a number of abstracts and a few papers that came out of these investigations. And some of the factors that people studied included the level of humidity in the incubators, the type of oil, and the oil has different properties like the density and the viscosity. The initial humidity of the oil, this is kind of interesting. So people thought maybe if I put the oil initially into a humid environment and then transfer it to the non-humid environment, that would somehow reduce this problem we're seeing. In fact, that turned out not to be the case. The volume of drops and the associated properties associated with the volume, including the amount of surface area associated with that, it was also investigated and shown to be a contributing factor. And also the thickness of the oil that is used to cover these drops. And all of these factors independently in these studies were shown to have an effect. And our company, Cook Medical, makes incubators, and we also make culture media. And so when these ideas began to sort of percolate in the field, we began to think about this and how that relates to what we're doing as a company. And so they approached me as one of the scientists in the IVF arena to think about this and to try and figure out how big of a problem I really felt this was. And so, of course, the first thing I did was I looked at some of the literature and began to assess what people have done. And then I wanted to sort of convince myself that some of these effects that people were seeing were true. And one of the reasons I did this is because a lot of these reports came out only in abstract form. And so there were a lot of details missing in the reports. And so the first thing I did was I essentially recreated some of these experiments in my own hands to see if I could determine how big of an effect some of these different factors had on the change in osmolality. And so the first study I did was I looked at the effect of the size of these drops used in the culture and how that affected the change in osmolality. And so the way I set up these experiments is I would start at time 0 and I would set up a system, and in this particular case, different dishes with different drops of oil. And I standardized the volume of, different drops of media. And I standardized the volume of oil in this particular case to 3 milliliters. And so I wanted to look and see how much of an effect the different volumes of media had. And these are the data that came out of that. And so each of these time points here represents the osmolality measured on different days. And so what I did was then I just performed a simple linear regression to determine the rate of osmolality loss over this time period. And what was interesting in this particular study is I noticed that it looks like there's a difference between the 30 microliter volume of medium versus the other two. And you can see when you perform a 95% confidence interval for the average of these, that appears to be the case. There's a significant overlap between these two averages where the 30 microliter volume had a much higher rate of water loss. And that was a bit contradictory to what had been shown in one of the other reports. And so I began to ask the question, why would that be? Why would I not see a difference between these two? And with these microdrop systems, the surface of this drop forms sort of a dome shape. And I'm not sure you can see it too well here, but if you look at a side view of these drops, you can see it forms this dome shape. And so there's a surface area that's associated with the different volumes of these drops. And so initially I tried to understand what that surface area would be and try to estimate what that would be. And before I went on to this more sophisticated analysis approach here, I tried to estimate that simply by modeling them either as a hemisphere or an obloid spheroid. And I could never get very good estimates, in my opinion, at that point of the surface area approach. And so then I was informed of this microscope we have in an engineering testing laboratory at Cook, which would allow you to take side views of different objects. And so typically in the IVF lab, you would be looking at things either from directly below or directly above. And I knew what I needed to do was I needed to look at these things from the side to get an estimate of the height of these drops and the overall surface. And so by taking this approach and then performing some quantitative image analysis and then doing some calculations, I was able to estimate the surface area of these drops for the different drop sizes. And to make a long story short, what turns out to be very interesting and explanatory, in my opinion, of the last set of data is the ratio of the surface area to volume decreases as you go from the small drops to the large drops. But what's interesting is the surface area to volume ratio estimates for the 40 and 50 microliter drops are identical. And I think that explains why what I was seeing earlier in terms of the water loss rate from the 40 to 50 microliter drops being the same. I suspect that's because the surface area to volume ratio is the same for these drops. And instead of using either the volume of the drop, well, instead of using the volume of the drop as the fundamental variable, I use the surface area to volume ratio of the media for the rest of my study, because I think that is a better predictor of this phenomenon. And so then I went on to detect the effect of different oils that are sold in the IVF market on these osmolality changes. I'm sorry. And so I acquired 9 different oil types from 7 manufacturers and began to look at the effect of these different oils on this phenomenon. And I used the other parameters, which I show here. And so what I determined was each of these just represents different oils categorized. And so there is quite a big difference between the types of oil. And if you, again, perform an analysis just looking at the estimate and a confidence interval range for the slope of these, you can see there's quite a big difference. Now, I had previously performed experiments under similar conditions, but in a humidified incubator. And the expected rate of change in the humidified incubator is notably lower than any of these particular oils. Now, I don't like to use things like brand of oil as a fundamental parameter in a scientific sense because it's really not very scientific. So then what I went on to do with the help of one of my colleagues at Cook Research up in West Lafayette is we took these oils and we measured the density and the viscosity of these oils. And the data for those are shown here. And so what these show is that there's quite a broad range of viscosities and densities for these different oils. And those specific data are shown here. And there is a relationship between these parameters, the viscosity and this change in osmolality from what I just showed you. And so I figured I better choose one or the other of these because they're fairly tightly correlated. And I decided to choose the density as the parameter of the oil instead of viscosity, primarily because it's a better fit to the data. A linear fits, it's a tighter fit to the data, and it's much easier to measure. Measuring density is a lot easier to measure than viscosity. And so I decided that oil density would be another parameter I would include as my studies progressed. Okay. And finally, I began to look at the amount of oil used in these systems. And oftentimes in the past, people have used the volume of oil because you'll take a dish, which is like a cylinder, and you'll add a certain amount of oil to it. And so I did something similar to, again, repeat what's been done in literature to convince myself there's an effect. And I did see an effect, of course. As the volume of oil gets higher, the rate of osmolality change decreases. and it's a nonlinear effect apparently. And it apparently also seems like there's sort of a tailing off of the loss of osmolality. And this can be explained, as I'll show in a bigger study, that as you increase the amount of oil, you're getting a higher and higher, essentially buffering volume of oil above this drop. Now, in this particular case, I started using a format for the medium that did not include a drop geometry. It was in a small well. And so I included 40 versus 50 microliters drops in this study too. And it was only because it was easy to do, not because I was looking at that independently. But nevertheless, it looks like the amount of oil used is also contributing significantly to this issue. And so having done all these preliminary experiments and now sort of intuitively figuring that all of these different factors would interact at least to some degree on this phenomenon, I went ahead and began to dig into how to perform a multi-factor response surface type study using these different variables. And so it just happens that someone at the company, which is a Cook company that I used to work for, that now is part of Cook Medical, had acquired a site license for Design Expert. And so I began to tinker with the software and found that I really thought it was intuitive and quite easy to use and quite powerful. And I invested in all the books that they publish, which are quite not only informative, but quite entertaining to read. So any of you who are interested in this, I would recommend getting those books. But sort of being a novice to this, I found it quite easy to use Design Expert to help me through this problem. So I chose the variables of surface area to volume for the media, the density of oil across this range here, and the thickness of oil over the media itself as the three factors that I would look at in this multi-factor RSM study. And I chose these ranges for a couple different reasons. One is practicality. This is the range of densities that can be obtained from people who sell oil into the IVF industry. Surface area to volume, this would represent a range that I expect at least would cover the surface area to volume ranges one would expect to experience in the different ways in which people culture embryos. And the same can be said for the oil thickness. So I started off with the face-centered central composite design and use Design Expert to help me with this design. And then after this first set of data was collected, and I continue to augment the data using different factor level combinations to get a total final experiment. And I didn't do all of that before I did the analysis. Of course, I performed the first block of experiments, and I would look at the data and determine what it looked like, if I was on the right track, for example, and where some holes in the experimental parameters would be, and then just use the software to help me augment this design. And finally, I ended up having 107 independent combinations of these three factors in 5 different blocks with 19 to 24 runs per block. And the software does a great job of randomly ordering the factor level combinations within the block. And also, when I was using this 96 well plate here, each column of wells would have included one treatment. And so not only did I randomize the location within this plate, I also randomized the order in which I sampled these 8 wells. And for each of the wells, I would take a sample, measure the osmolality with my osmometer, record the time it was measured and the osmolality itself. And then in the end, I ended up with a data set with the osmolality change over 7 days, and use initially a linear regression analysis, like I mentioned earlier, to determine the rate of change in osmolality under these various conditions. And so in all the conditions, it appeared that a linear model over the 7-day period fit these data quite well. And so I used a slope parameter, which came out of the linear regression as the final parameter to input into the Design Expert file and use that as the endpoint. So then what I ended up doing is going through the process that is laid out in Design Expert on how to analyze models. I looked at the quadratic, initially the quadratic, cubic, and quartic model, and I used some of the guidelines that are laid out in the software. And it turns out when I looked at the quadratic model, which fit the data pretty well, it turns out it didn't fit the data well at some of the extremes within the data set. And when I moved to the cubic model, then that cubic model allowed the data a better fit in those particular areas. And moving to a quartic model had very little additional effect. And so I felt like the quartic model was really an overfit of the data, but the cubic model was a better fit. And I'll explain in some of the graphs in a couple of slides here where that better fit occurred. And so using these criteria like AICC and BIC and the others, I was able to conclude that a cubic model would be better than the quadratic model. And then I did the backward subtraction effect of some of the terms within the cubic model to end up with the modified cubic model that I stuck with. And it's just as a note, I had to perform some transformations during this process to make the data better fit the model assumptions. And so in the end, I ended up with a truncated cubic model with the terms shown here. All the terms are highly significant. In fact, all of them are less than 0.0001 with the exception of B, but that's still very significant there. And so I ended up with this final model. And then I went through the process, during this process of selecting the model, looking at the different graphs that Design Expert allows you to look at, the predicted residuals, the residuals versus run, the normal probability plot, as well as the predicted versus actual residuals here. All of them, once I performed this transformation, looked really good. I didn't see anything that caused me any concerns about the model fit to the data. And so finally, after all this process, I ended up with the model as shown here, where the osmolality change per hour, or change in osmolality over time, can be described well by this particular truncated cubic model, where the different factors include the surface area to volume, the oil height, and the oil density. And so when we look at some of the graphics that Design Expert provides, we can see how well the model fits the data. And you can also see how the data points and the output variables change in different sections within the design space. And so what we see is, and these are in the coded variables here, as the surface area to volume ratio gets higher and as the oil height gets lower, there's a dramatic rise in the rate at which osmolality changes. And these three graphs represent different levels of the third factor, which is the density of oil. So as the oil density is lowest and the surface area to volume gets higher and the oil height gets lower, you have a very dramatic effect on the rate at which water evaporates from these media drops. In fact, it's so dramatic, it's greater than 1 milliosmol per hour, which is quite striking. And so for every hour this dish is in culture, the osmolality is increasing by 1 milliosmol, and that's pretty strong. Now, as you look at these two graphs on the right, you can see that increasing the density of oil does augment this problem to some degree. And it really only has its effect in the extreme section of this design space. And so as you increase the oil density, the rate at which the osmolality increases in this extreme section here drops. But it has not very strong effect in the middle part and the leftmost part of these areas in the design space. And so this really gives you the opportunity to sort of think about how you might tailor an embryo culture system and what changes you might want to make and what changes wouldn't necessarily need to be made to decrease the rate of this osmolality change. And just as one example to help you understand that. And so if you notice the oil height here, if you look at the middle where the oil height is 3 millimeters, you can see the slope of this gradually rises as you go from a low to a high surface area to volume ratio. But that doesn't change very much as you increase the thickness of the oil. And oil is a rather expensive component in embryo culture systems, and so you don't want to waste any of it. So what this type of analysis would tell you is once you get to approximately 3 millimeters of oil thickness over your drops, there's really no benefit to increasing that and adding more oil to your system. In fact, there are means by which more oil can be detrimental, but it doesn't have to do with this particular problem. Okay. So once I had all of this completed, I went back and I chose 21 independent design points to look at and determine experimentally what the change in osmolality was, and then use the model to predict what the change in osmolality would be expected to, what that change is expected to be if the model is accurate. And what this graph shows is for all of those 21 points, the actually experimentally measured change is shown with the red dots, and the predicted rate of change in the confidence intervals is shown by these 95% confidence interval bars. And in all cases, the model predicted with 95% confidence where that expected change would be. And so I was fairly confident that the model was pretty accurate in predicting what we would expect to have for an osmolality change under many different scenarios within an embryo culture system. And so finally, I'd like to end by discussing in a real world setting what actually people experience under these scenarios, and then show how the model would actually accurately predict this. So if we go back to that paper I mentioned at the beginning of the talk, where this group set up an experiment and cultured embryos in a humidified and a non-humidified setting, I wanted to see what the actual numbers would be for osmolality change over time, which unfortunately they did not report in the original paper. And so I went ahead and got all the details from that paper and got the equipment and supplies I would need to repeat that. And so I essentially set up a replicate experiment from what they reported in their paper, and then determined this osmolality change over time, as I mentioned throughout this talk. And what the data showed is that there is a very dramatic difference in osmolality at the end of the 7-day period between culturing in a relatively dry state, shown by the red dots, versus a relatively humid state, shown by the blue dots. And as far as a percent change from the beginning, when you culture these in the humidified setting, and this is 73% relative humidity, it's a little bit lower than what most incubators achieve. And that's because of the way I had to modify the incubator to execute this experiment. But even so, in the humidified environment, the osmolality changes by only 3.5%. Whereas in the non-humidified setting, it changes by over 13%. And that's a pretty big change when you're culturing embryos that are so important for people. And so sort of to look at the model, and this is a data set that's independent of the data set I showed previously. The actual change in osmolality under the non-humidified conditions was 0.208 milliosmolal per hour. The model predicted on average it would be 0.223 with a confidence interval of 0.162 and 0.292. So this actual experimentally determined osmolality change fit pretty well within the 95% confidence level of this of the model predictions. Okay. And so I see that I've only taken about half the time, so maybe this will stir some discussion. But this kind of change is something that in the clinical setting, you kind of hope you don't have, because there was a pretty significant assumption made at the beginning of when people began to use these non-humidified incubators. And that was, as I mentioned, that the oil layer would essentially protect the drops from significant evaporation. But what I and others have shown is that's in fact not the case. And particularly, it's not the case under certain conditions of embryo culture. And that's something that people have become quite aware of, and people are seriously starting to consider whether there should be any circumstances when culturing in a non-humidified incubator is appropriate. But what I tried to do with this particular study is take a set of several variables that people in the community believe are having an effect and put those into a multi-factor model, a multi-factor experiment that I could use to develop a predictive model like this. And then hopefully the community will have that information and be able to sort of perform a what-if analysis as they're trying to refine their embryo culture methods, and use this information to try and predict what would be better methods to culture embryos under perhaps limitations that they have to work with within their laboratory. And so I'm really hoping not only is this informative, but it's actually useful for the community as a whole. It will help guide future practices in terms of improving embryo culture. So I think I'm going to end the talk there and just thank a bunch of people, some within Cook Medical and others without, that include individuals in my specific laboratory, which are shown here. I have a good friend, Dr. James Benson, who is a mathematics and biology professor at Saskatchewan, and he helped me with this surface area measurement process when I was working through that. AJ McKenchny is a statistician at Cook Medical, and he sort of gave me some confidence at the beginning when I wasn't quite sure I knew what I was doing with the RSM and experimental design, and he let me know that it looked like I was on the right track. And then one of my good friends, Brian Woodard, who's the director of global product development with reproductive health, who helps me out with quite a few things not laboratory related. And my director, Elizabeth Brown, who's one of the global directors for R&D at Cook. So I'm going to leave it at that, and hopefully we can have some interesting questions that came out of this.
All right, thank you so much, Stephen. Personally, I found that whole thing very fascinating to hear about more about, you know, the background that goes into, you know, I'm going to call them the range finding tests, you know, that really essential fundamental subject matter knowledge that helps if you know that prior to jumping into a designed experiment. I think that's super important to take the time to do that. And you get more out of your DOE by doing that. So some comments and ideas here, which I think you're open to ideas. Since there is noise in estimating the slope parameters, did you consider taking the noise into account? Maybe the another response could be the root mean square error of the fit. It could be another response or some other approach.
Not directly. I understand. Yeah, I understand that. I understand the question. It's a good question, and it really begs the question in my mind, not being a statistician, is to what degree do you have to do that for the individual runs versus having a large design space where you select data within a bunch of different areas within this design space, and the model itself would sort of, if the model's accurate, you would think that the model would sort of filter a fair amount of that out. Now, there were a number of replica points that I conducted within this particular study, and I can't recall off the top of my head how many there were. But if I can still go up to it, what you notice is when we have replica points, which you can see a few here and here on these corners, the points themselves are pretty tight. There is more noise in the data when you get to some of the extremes, like we see out here on the far right. And my pointer's on the wrong screen. Sorry about that. I guess, can you see my pointer now, or it somehow changed back to a pointer, not a laser pointer. So let me do this.
Yeah, we can see it. Yep.
Okay. So there's more noise in the data when you get to these extremes, and that's probably not surprising. But you can see in several instances there are replica points like we see here and here, and in these two, and then down in the higher density of oil and the lower density of oil. And the noise in the data is pretty small at those points. And so what I noticed is that along this edge of this design space is where I saw the most noise. And in fact, I didn't mention it, but this is the area within the design space that the quadratic model wasn't fitting very well. And so it really took the cubic model to fit this rapid increase in slope we see here on the edge. And another thing I should say, and I try not to pretend I know everything because I don't, but it would be interesting for me to understand a bit more about how I would do what was suggested in the comment and take the mean squared error, for example, into account.
Okay, another comment. Stephen, there are some approaches that allow the response to be a curve or a line rather than a point. Functional data analysis is one such approach I have seen in R and JUMP. I'm
Okay.
not sure you would learn anything more, but it might be interesting to try. Thanks for
Okay.
the great example. That's from Arvid.
Sure.
Arvid Singh.
Can anybody explain to me what functional data analysis means?
Um,
I might just give people somebody time to type it in, and then you can come back to that.
yeah, and if any of the presenters on the line, you guys can unmute yourself, and if you have a comment, that's fine. You're welcome to do so.
Yeah.
In the meantime, I will. Oh, okay. Hold on. Let me see if I can unmute Arvid here. Okay, Arvid, you should be able to unmute yourself now and speak.
Okay, can you hear me?
Yep.
I can. Yeah.
Yeah, well, functional data analysis is about the whole idea is I don't have the response is not a point, it's a function. And you
Sure.
can actually build in to the modeling part parameters like a slope would be an example, but you actually are modeling not just a point, you're modeling the whole thing. So
Yep.
slopes and quadratics and even exponentials and other things can get taken into account. Some of the approaches use spline fitting and other type things to do that, but it's excellent for predicting in situations like you're describing.
Okay. I'm gonna make sure I write this down and see if I can get some background on this. But yeah, thank you for your comment. I'm always open to this because, as I mentioned at the beginning, I'm not a statistician, but it's something I'm really fascinated by, and I try to learn as much as I can. But I'm sure there are people who know a lot more than I do out there.
We're continuing to learn. Been doing this for 33 plus years, and I'm still learning.
Okay. Well, I won't feel so bad.
Thanks, Arvid. Appreciate your comments. And as an additional comment on that from John Davies, he says that functional data analysis is used when the response of interest is a functional form rather than a scalar. It's like
Okay.
principal components, but for responses with curves.
Okay. Okay.
Okay. And John also had a question. How did you vary the surface area to volume ratio? Was it just by changing the volume of liquid in the cup with the surface area of liquid to oil interface remaining constant?
That's exactly how I did it. In fact,
Okay.
in the drop setting, the surface area is a function of the volume. And so when you're in a dish, and I'll see if you can still see my screen.
Yep.
A dish like this has real small wells, and the wells are cylinders. And so essentially you have a fixed surface area. And so the more volume you add, or the less volume you add, you change that ratio. And so that's that's the means by which I change that ratio. And what's interesting is, whenever you go from a drop setting to a well setting, people think, well, it's not the same. And so the data doesn't really have any meaning. But the truth of the matter is the physics behind it is the same. And I sort of, hopefully anyway, I convince people that that is true by the experiment I did earlier, where I showed that it's the surface area to volume ratio of the drop and not the drop volume. That is what is driving the loss of osmolality, the rate of loss of osmolality. Yeah.
Yeah. Interesting from a science standpoint, very much. And we've got a lot of power users in on our talk. So Michael Bradshaw dropped the Wikipedia reference for functional data analysis for as much as it can be trusted.
There you go. There you go. Okay.
Always a good first place to go.
Yeah, well, it's a good first place. That's right.
All right. Do we have any other comments or questions out there?
Even if people have just general questions about IVF and assisted reproduction, I'm happy to answer those. That's often something people are curious about. One thing I will mention, and this is a huge gap in our knowledge, is that people have performed these studies and nobody, this is unfortunate, but when you get to the end of this, you say, well, I can predict the rate of osmolality change under various scenarios. But the question is, so what does that mean as far as my embryo health? Under
Mhm.
this scenario, like if we generally think less osmolality loss is better, but is there a minimum below which it doesn't matter? And that nobody knows. And so what I want to do is go back to a lot of these papers and abstracts and see if I can extract all of the information I need to use my model when the paper abstract also has embryo development and pregnancy data.
Mhm.
The frustrating part is so many of these publications were only in abstracts, and so they don't have nearly enough detail for me to do that. And I may just have to try and call everybody and see if they're willing to share that information with me.
Yeah,
But
track down original authors, and
yeah,
I
but
assume a lot of this is done in the laboratory, and then tracking it all the way through, like you said, to the final, like a live birth, and the
yep,
health of that child.
that's really
Um,
difficult. Yep. But at least to get some, generally speaking, if the cohort of embryos shows stress, then the developmental rate will be reduced. And that at least gives you an initial point to start with. We would expect that if the conditions are better, the embryo development rate would be better. And so at
mhm.
least we can start there.
Mm. You mentioned that the oil was expensive. Is the oil cheap or expensive compared to the biological materials?
Oh, much, much cheaper
Yeah.
for sure. But you know what's crazy on our field is I suppose it's like everywhere. People tend to sort of, what is it called? They sort of nickel and dime themselves to death. But it's the opposite of that, I guess. They worry about expense when you know expense in that particular case shouldn't be worried about.
Hmm.
Um, and so yeah, it's expensive. The other thing is, despite all the quality control that goes on, mineral oil tends to be a bit toxic to embryos. And once in a while, this toxicity won't be detected, and you'll get a batch of embryos that doesn't develop as well as you'd expect because there's some toxic factor in the oil.
Huh.
And so I'm assuming that the more oil you put in your dish, the more this stuff is going to diffuse into the media and affect your embryos. And so I would think you'd want to know what is the minimum amount of oil to use, not just for a cost standpoint, but for potentially reducing toxicity on the embryo.